Hey guys, today we're going to learn about karyotyping and how to interpret a karyotype. So what is a karyotype? It's a map, or more accurately, a picture of your 23 pairs of chromosomes, or your 23 homologous chromosomes. They're arranged from longest to shortest. The first 22 pairs are called your autosomes. And those autosomes are going to include genes for traits such as hair color, your height, whether your earlobes are attached, and those types of things. The last pair, although they are not the shortest, the last pair is going to determine your sex. They're called your sex chromosomes. And they will also include genes for traits such as hemophilia, color blindness, and baldness. So now that we kind of know what we're looking at when we see a karyotype, let's talk about how you make a karyotype. First thing you're going to do is obtain a sample of cells. Okay, we oftentimes use karyotyping to diagnose diseases in embryos before a baby is born. So we'll often use a really long needle and stick it into the abdomen of the mother and extract some of the fetal cells. We'll then add a chemical called colchicine to those cells, and that medicine actually causes the DNA to coil up and condense into chromosomes so that we can see the DNA better. And once we have that DNA fully coiled up, we'll take a picture of it, and then we can rearrange and separate the chromosomes based off the length and match it up with its pair so that we can form the picture, our karyotype. When you look at a karyotype, they're going to look like one of these two on the board. The one on the left is a female karyotype. And I know it's a female karyotype because the autosomes are all there. But then the last pair, the sex chromosomes, there are two chromosomes listed here above the X, meaning that this person has the genotype XX, which makes her female. All pairs are the same length. Okay, I know some of them look bent, but that's okay. Still, they're still, it doesn't matter if they're bent, they still have the same genes on them. Okay, they just didn't line up straight when we added that chemical. Now, the one on the right is a male karyotype, and I know that because when I look down here at the sex chromosomes, there is one long and one short, an X and a Y. And so he has his autosomes being all the same length, but actually has a different pair of sex chromosomes, which makes him male. Now, things can go wrong during meiosis, and when we incorrectly separate chromosomes during either meiosis 1 or meiosis 2 in anaphase, a process known as non-disjunction occurs. And the gametes formed from that cell division either have too many chromosomes or not enough chromosomes. And depending upon the chromosomes that we have either too many or too few of, we can have one of several different chromosomal genetic diseases. So the three diseases you need to be able to determine that a person has are Down syndrome, Turner syndrome, and Klinefelter syndrome. You can determine a person having Down syndrome if they have trisomy 21. And trisomy means that they have an extra 21st chromosome. So when you look at those autosomes and you see that there is an extra of the 21st pair, you know they have Down syndrome. If you're looking at a karyotype and you look and they have all the autosomes the same, but they have a monosomy or only one sex chromosome, that person suffers from Turner syndrome. And if you're looking at your karyotype and they have the autosomes all the same, but then they have a trisomy for their sex chromosome and they have an extra X chromosome, then that person suffers from Klinefelter syndrome. So let's take a minute and watch this short little clip.
Do you know anyone with Down syndrome or Klinefelter syndrome? These are examples of chromosomal disorders which are due to non-disjunction. When non-disjunction occurs during meiosis I, replicated homologous chromosomes do not separate correctly. One daughter cell retains both replicated chromosomes. The other daughter cell lacks that chromosome. When a haploid sperm fertilizes an egg with an extra chromosome, trisomy occurs. When a sperm cell fertilizes an egg that lacks a chromosome, monosomy results. In this scenario, either a trisomy or a monosomy results. Non-disjunction can also occur during meiosis II. In this scenario, meiosis I occurs normally. When the daughter cells separate during meiosis II, one daughter cell divides normally, but the other daughter cell divides abnormally. One egg lacks a chromosome while the other egg has an extra chromosome. When a haploid sperm cell fertilizes a haploid egg, a normal diploid results. If the sperm cell fertilizes the egg that lacks a chromosome, then monosomy results. If the sperm cell fertilizes the egg with an extra chromosome, then trisomy results. In this scenario, there is a 50% chance of a normal diploid, a 25% chance of a monosomy, and a 25% chance of a trisomy.
So let's now look at interpreting karyotypes. So for all of these karyotypes, we want to identify the sex of the individual and the disease that they have if they have one. So take a minute and look at the first karyotype over here on the left. The sex of the person is going to be, they have two of the same sex chromosomes, so they are going to be female. And if you look, they also have 3 21st chromosomes, so they are female with Down syndrome. Okay, take a minute and do the next one. So when trying to figure this one out, you'll notice that they have all the autosomes paired up appropriately, but they only have one X chromosome. So that means that they have Turner syndrome, and since we have no second sex chromosome, the default sex in humans is female. So even if you only have one sex chromosome, it must be an X, and you will be female. So this person would be female with Turner syndrome. Let's try the next two. The one on the left, we're going through and we notice that, oh, there's a trisomy there. But then when we look here, oh, there's an X and a Y. So this person would be male, and they have Down syndrome. For the last karyotype, you might have said that person would be female and have Turner's. But that's where you need to go ahead and you actually need to, instead of relying on the chromosomes being numbered, you need to go ahead and actually count them out. Because if you count them out, you will notice that there are 23 pairs and an extra, which means that it's not this person is not female with Turner's. This person actually has Klinefelter syndrome. And if you have even if you have 2 X chromosomes and a Y chromosome, if you have one Y chromosome, you develop into a male. So a person who has Klinefelter's will only ever be male. They will never ever be female. Okay, then go ahead and answer these 4 questions. What's the name for chromosomes paired 1 through 22? How does a female karyotype differ from a male karyotype? what is non-disjunction, and which chromosomes incorrectly separated to cause Down syndrome.